I will be discussing with you, and hopefully you're not completely dead after all these lectures, about high-throughput biology. And from one side, it will be an introduction to the topic, because I remember that many of you wrote you're a biophysicist or computer scientist. And on the second place, it will be also regarding networks, a tiny little bit. And also, I'm very happy to be within these two talks, between Alina's talk on metagenomics and partly a network talk, because this is all very much related to the topic. So, to those who were not present here during the first lecture, I am a medical doctor switched to the scientific track. I was working with as a general practitioner during my postgraduate training in the Center for Medical Genetics, and since 2019, I am pursuing my PhD in systems biology and liver disease. Since the last year, I'm researching as a research associate at Medical University of Vienna, and this talk particularly, and let me just switch my pointer, will be regarding this first first paper I'm showing here a little bit. Just 1 second. Yeah, about the transcriptomic signatures. And do not worry too much. I will explain what does it mean just in a few moments. So if I want to tell you just 3 things today, it will be what's happening in the high-throughput biology and why is that the thing, which is our hitchhiker's toolbox of machine learning, and not only machine learning stuff we can use, and share my biomarker journey, which was just recently published. So let's go to what is happening. If you were a dedicated biology student, or maybe you were working in the lab, you know that there is a wide spectrum of conventional microbiology methods, which basically allow you to answer the question whether my specific DNA sequence is damaged or has a specific mutation, or whether my protein of interest is present in whatever sample used to measure that. As in here, for example, this is the Western blot, a method where you can detect proteins based on the immunoprecipitation, separate them based on their molecular weight, and can understand whether your protein of interest is A, present there, B, more present in your sample A versus sample B versus sample C, and so on. And of course, it's quite okay throughput, but not high throughput enough for today's challenges, although this method is still really widely used. And the second approach, which even those of you who didn't study biology can be aware of, is PCRs. And this is the example of the qPCR. So this so-called quantitative real-time quantitative polymerase chain reaction. Then basically we have product of some gene expression, so they're mRNAs, and we suspect that they're very important for us, important to the extent we want to measure them. Maybe it's because we want to measure how much of the hepatitis C virus RNA is present in a sample of patient, or we're simply interested in the function of certain gene. And based on specific targets, so that we know what we are looking at, when we have 2 to 5 to 8 to 10 genes of interest, we can just measure how active are they in whatever sample you have. Should it be your cells or your tissue or even your soil if you're working with metagenomics? Again, very important, still relevant, not high throughput. So what's happening? Modern biomedical research operates in a very large data sets. We need data sets to be explicit enough to answer all our questions and be accessible for reanalysis should we have any more questions. This is the big philosophy behind this high-throughput biology. Even though we still use conventional methods, they are not sufficient anymore to follow our tasks. And these high-throughput methods, which I will be discussing in a few moments, allow us to perform such so-called hypothesis-free or exploratory research. Think of a case when you have a patient with a new rare disease nobody ever described, or if you want to study new organisms or new plants or new bacteria. So you might not necessarily know what you're looking at, but you are aware that you will find something interesting if you look deep enough. And this is why we have these methods. And basically they answer, or their application, their acquisition can answer us a few questions, such as what are the new molecular targets we never explored before in a certain disease, whether we can find new biomarkers, new drug candidates from millions of compounds, and literally we can screen for that much nowadays. There are many examples of such high throughput data sets, and this is just a few of them. The first one on the left part of the slide is a high throughput screening readout. So this is some technology which is known as pharmacoscopy. It was discovered initially at our institution, now commercialized as a company where basically the guys in the company can screen for the cells of patients with leukemia and find the more suitable drug that can treat the specific type of leukemia based on the specific mutation in the cells, which not necessarily might be known already. And this is like an approach when we can, using also machine learning tools, analyze thousands of cells in the same time based on their phenotype after treatment, how the intensity of several marker changes, how the appearance of the cells changes, and so on, to find therapies which are the most suitable for the specific case in personalized medicine. The second one, I will be focusing on mostly through the talk, and this is called next generation sequencing. And if it's the first time you're meeting this word, please don't be afraid, because sequencing basically means that we are reading the sequence of whatever molecule we're interested in this case, DNA or RNA. And in a moment, I'll explain why does it look exactly like this. And the third case is the the proteins or metabolite spectrum of whatever you measure them in. Should be a patient's serum or soil or whatever other biological relevant environment you're looking at, where based on the spectral qualities of different proteins or metabolites, we can identify them and measure between different samples. And of course, you can get hundreds and thousands of them. That depends on your machines and the methods you're using. And before I switch forward, it's important to obtain this vocabulary, which is basically any word you can imagine in biology and in omics, which is in principle is an assembly or spectrum of something within a sample. And this something can be, for example, genomics, which is information about the structure of all genes, approximately all genes, in your sample or organism or environmental setup, whatever you're looking at. Transcriptomics, which meaning that all transcripts or all products of expression of a certain genes. Proteomics, meaning that all the proteins, lipidomics, all the lipids, and so on. There are many of them. I don't think there's a single vocabulary that can capture all the omics, but just for you to not be afraid of such words. And where the sample, meaning that the transcriptomics, for example, is a combination of all genes expression on sample, which can be a whole organism or a tissue or a cell or environmental setup in case you're working with soil or water or whatever. Now, when we are aware of that, let's focus on transcriptomics, because this is what the talk mostly about. And there was some development or evolution of these methods through the development of high-throughput biology. The first one, or not the first one, but actually the one which we still use nowadays very actively, is RNA sequencing. When you have your cells of interest or tissue of interest, this is very frequent application nowadays, and you want to quantify what is the activity of all the genes or specific genes of your interest, for example, in this sample. You use specific kits to isolate RNA from the samples. You fragment them to various short segments, which is usually done by so-called wet lab biologists or biologists working in a biological laboratory. We convert them from RNA fragments to the complementary cDNA fragments for better stability. Mark them with specific ends that will be important for the machine which will read their sequence, and finally perform the sequencing itself. And for data science perspective, that gives us the following results when they have different exons or protein coding elements of a specific gene, and the number of elements which were produced from this specific exon. And we can quantify them and compare. For example, whether gene A is more active than gene B in a specific sample, or whether gene A is more active than gene A in sample A than in sample B. So from the data science perspective, these are the questions which we can answer. And on the right side, there's a very typical biomedical application of that. When we have certain tissue, imagine a cancer in a patient, and we make biopsy of this tissue, capturing all the cells repertoire which can be there. We homogenize that, receiving all the RNA which was inside, and we sequence it. So we can compare different samples between different patients in this way. From the data science problem, because it's a data science school, we should convert it always in this format. We can imagine that as a matrix where we have genes and samples, as in this case, and you can compare whether your genes are different or stable between different samples, and which genes, and so on. Important to mention that usually you're working with thousands of genes, like from the standard RNA sequencing analysis, you get around 20,000 genes readouts over the number of samples you can allow yourself to sequence. So that really depends on your experiment. And these are types of questions you usually can answer with this method, like which sample has the highest expression of a certain gene of interest. If you compare samples in different groups, what can be the most differentially expressed genes, how we can group these genes, whether they're functionally important or not. So these are the types of questions you can answer either with basic statistics or with machine learning method of your choice. There are many approaches and not many gold standards in this field. However, it's not always that simple. This is the second part of the figure I presented you on the previous slide, where instead of taking the just tissue of your patient or even if you work with soil, whatever was in the soil, but actually splitting this tissue or soil microbiota for separate cells and sequencing all the RNAs in this each separate cell independently. And in this case, if you're imagining data matrix, you can think of something quite more complex because now we have similar data frames or matrices, except instead of samples, we have cells within the samples, and we have many such matrices to analyze. And of course, with this increased modality of our data set, there are more potential applications for machine learning or more advanced statistical methods, but also there are much more questions we can answer. For example, if you're working with cancer and you're interested in whether your malignant cells are the same between two similarly classified types of cancer. Is it true or not? Whether they're functionally doing the same or not? So this is the type of questions you can answer. And of course, because we have this separated to different cell types, we can assign identities to our cells based on the genes expressed. For example, if the cell A, cell 1 expresses the genes which are specific to fibroblasts, then we can assign this identity and separate it from non-fibroblast cells based on the methods we will discuss in a few moments. And also you can use this, I didn't include it in the presentation for the time's sake, but you can use it with the previous method, so the bulk RNA sequencing or the sequencing of the whole tissue, where you can make probabilistic deconvolution or probabilistically try to understand which was the cell composition of your tissue that is sequenced based on this reference single cell data set. And many people use it nowadays for economic reasons, but also just to go back to your data sets which you previously done and try to understand which cell composition was there, again, based on the probabilistic models and not on the real biology, but still. But sometimes it can even be more complex. Like, for example, in this case, when we have the same data set as in the previous slide, but also for each sample or even each cell, we can have different additional modalities, such as additional protein readouts. So we can have all the spectrum of gene expression for each cells, but also the protein concentrations or metabolic concentrations, or we can have functional assays, including imaging. Remember the first slides where I showed the high-throughput screening and the cells microscopy. So imagine that for each cell you also have the sequencing data. And as a data scientist in bioinformatics, you would need to think which machine learning or statistical methods to apply here to make some sense of that, because ultimately this is why we're doing this type of experiments, to make some biological conclusions. And of course, there's a whole lot of different questions, such as whether like how to integrate data from these all modalities for each cell. How do we normalize that? How do we prioritize the markers, and so on. So ending this introductory part, I would say there are two big trends now in high-throughput biology. The first trend is that people apparently are interested not only in what is happening, but also where it's happening. And on the top portion of the slide, you can see two images. So on the left one, it's a simple histological image, which departments of pathology in hospitals acquiring all the time. And on the right side, it's so-called spatial biology, the spatial transcriptomics image, when we have sequencing of cells which were located on this slide, and we also know where the sequencing happened. So we can backtrack, for example, and see that some cells in this portion of the slide above here are different from the ones which are below here. And this also can lead to different biological insights. And the second big trend, which is quite long-lasting, but still very relevant nowadays, is multiple modalities. So something we discussed on the last slide, where actually we're interested in combining our insights and see whether my RNA products are being also found on the protein level, whether it is related to any features of the cells based on macroscopy and so on. So here we come to a very quick overview of the hitchhiker's toolbox, or the things which you can use as a data scientist, also from the machine learning perspective. And first, this is the standard workflow, very simplified, but we'll give you some idea how does it work. So when you have this old data set you acquired with such methods, you can filter out what you don't need. For example, genes which are not expressed with 0 everywhere. You can normalize the data and scale. For some methods needed, some methods do not need it. For example, if you want to compare something between samples, you obviously scale that. And then starts what they call the wild west of the high-throughput biology, because there is not much gold standards. And usually you want to reduce the dimensionality of your data set, because if you imagine that you have 100 cells and 20,000 genes expressed, it's a very high dimensional data set that you cannot really interpret without dimensionality reduction. And then perform whatever advanced biology tasks your colleagues from the lab are asking you to do. For example, whether your cells from the right side, from the right cluster, are communicating with left cluster, or other tasks. So for dimensionality reduction, it's actually quite a big topic, and the topic I personally like a lot, because it allows you to visualize your data and make it human interpretable, even for biologists or those who are not involved so much into data science. And you can see here 3 different methods. Some of you might know about them, some of you might not. The principal component analysis, which is showing the relation, the linear relations between your data points, in this case cells or tissue samples or whatever, in this case they are cells, into the space. So this is being calculated based on the basic statistical methods, no machine learning involved. Or something more advanced, which also captures nonlinearity, such as, for example, UMAP, or uniform manifold approximation and projection, where complex nonlinear relationship, which I will show on the next slide, can be captured and plotted in the way that all this distance is actually meaningful and allow you to distinguish the cells of your interest or see how closely they're being grouped. And for example, the third example of this method is de-distributed stochastic neighborhood embedding, just another machine learning based approach that allow you to reduce dimensionality of your data set. Now this sounds very complex, but in reality you can apply whatever method you want, not even in biology context, but also in any other. For example, there is a ready to use UMAP learn package to make UMAP dimensionality reduction on whatever data you need. And this is how it works. UMAP is an unsupervised method that allows you to play a lot with your dimensionality to find the best 2D embeddings for your cells or whatever data points of interest you're having. So first, it tries to understand how the cells or whatever data points you're having in your data set are related to each other in the multiple dimensions, so approximating this between them, and then it projects it to two-dimensional space, moving them around each other, trying to recapture all these multiple dimensions embeddings it just learned, and putting them in the most optimal position. Now the problem is there is no better or worse dimensionality reduction technique, which also opens you quite a lot of space for developing your own new approaches. This is what is called a benchmark for dimensionality reduction methods. On the left, you can see this beautiful 3D mammoth picture, and basically below that, you can see how different dimensionality reduction methods are successful or not successful in recapturing this mammoth in a 2D space. And that's the big problem, because if you work with single cell biology, in one organ or in another organ, different dimensionality reduction techniques might be the better or worse in recapturing your biology. And there's a lot of these techniques being published like every year. Some of them based on the more smart machine learning, some of them based on the simple statistical methods. So if you're happy with studying dimensions and embeddings and manifolds, you might find an application for your skills in this field, just working with a friendly biologist and trying to make the most sense of their data set. Basically, the only two questions to keep in mind when developing such methods is the method good at preserving local geometry, meaning that, for example, whether the horns of the mammoth will be recaptured also as the horns close to each other into this space, and B, is the method good at preserving global geometry, so whether the right leg will be far apart from the left leg, for example, or the head from the leg, and so on. For example, in my data set, one specific method works very nicely, but your map is not that nice. So this is something for you to keep in mind should you be interested in that. And this is just a very peak of the iceberg we just touched, because so the upper part is something I just told to you. So where we have this raw data, we normalize it, we apply different dimensionality reduction, we have some insights, but also there are totally different approaches for that based on machine learning. For example, the one which I personally like is called SCVI, which doesn't require you to normalize the data or scale it or whatever. It just uses a variational and autoencoders to make sense of this raw data, to learn its high dimensional representation and then try to recapture it in two-dimensional space. And this is a totally different approach. So if you're, let's say, trying to establish a new machine learning method for your non-biological data set, it's always a good idea to try it also, whether it makes sense in biological applications or not. For the sake of time, just jump into the last chapter, which is a new biomarker journey. I used one specific trick, this machine learning in the network context, to understand which gene markers can be related to the condition of my interest, which in this case is liver fibrosis and portal hypertension. So those who were attending the first talk already know what is portal hypertension, but let's say these are two conditions accompanying chronic liver diseases, and this is what we as a lab are interested in. So we had mice model of liver fibrosis, two different models for what it takes here, with two different chemical inductive liver fibrosis and chronic liver disease. And what we did, we built a time course experiment, and we had like 0 time point or healthy animal without induction, then animals at the peak of the fibrosis induction, and then animals at the regression stages when we stopped inducing fibrosis and just allowed them to spontaneously regress for a certain time. And at each of them, we actually acquired the transcriptomes. So we took the livers of these animals, we did the measurements we needed to measure, and also extracted and studied all the RNA products at all of these experimental time points. When I thought that we did additional measurements, it's important because as one of the subtasks for our study was to link gene expression to these external measurements, which we're interested in, such as the pressure in the portal vein, the amount of fibrosis animal has, and the liver injury. So the bottom part you don't have to see right now because I will jump to that in a few slides, but just to recapture what you already know and what I already showed, this was the raw data, so 20,000 genes, 48 samples, just as a count matrix, and basically as every transcriptomic journey, I started here. And to link my gene expression to the external markers, I use something which is super widely used, and it's not based on machine learning technique, which is called weighted gene co-expression network analysis, or if you try to convert this smart word, this smart phrase to the simple terms, meaning that find the genes which are highly correlating with each other, and within these groups, find those groups which are correlating with your external marker or measurement. In my case, I found certain number of groups of genes correlating between each other and correlated them to the presence of liver cirrhosis, portal pressure, abundance of fibrosis, or liver injury markers. As you can see, some of them are highly correlating, so we know these genes might be linked to these conditions. Then, converting that to the network problem, I tried to do calculation of 3 metrics. First of all, the gene importance, meaning that how much the gene is perturbed compared to the baseline in either the peak of the fibrosis induction, which I showed, or in the regression, so whether it returns back to the baseline in a healthy or not. And I've seen that you can see this strong negative correlation that actually the genes which are being induced by fibrosis also being reversed partly in the normal condition, but not every of them. So there is a part of genes which are showing more complex behavior. I applied a few network scoring methods, which I will show on the next slide. And on that, I will apply machine learning, which I will also explain on the next slide. So this is my network and how I built that. The middle layer was actually the first layer, and these are the models which are correlating with external features, such as portal pressure, fibrosis, and so on. So you can see 4 of them because I correlated them to portal pressure, fibrosis, the peak of the induction, and the regression. And the links between them were correlation of gene versus gene, so how genes are being co-expressed together. Having that, we can also think and infer transcriptional factors that are regulating them, meaning that if if 5 genes which have been regulated by a single transcriptional factor are important on this graph, the transcriptional factor is also being important. But also having these genes and knowing each protein that is being encoded by the genes, you can build a protein-protein interaction network using public database. And the machine learning approach, which I was referring to, is called random walk, and it's quite frequently being used in networks. When you start at a certain point, in my case, it was this bottom point, so gene regulatory level. You start at any node of this point, and your random walk goes to different nodes connected to each other until it stops. And this stopping of the network is being declared by first the constant you're setting in your random walks, but second also by the metrics you're providing to those random walks. And in my case, these were biology relevant metrics, meaning that how much transcriptional factor regulates certain genes and how certain genes are connected to each other or to the proteins of interest. Then you prioritize or cut off the threshold you need and resulting in some hub genes, which are the most important for your whatever external measurements you want to prioritize them. And also we validated them in human data sets. So these hub genes are useless unless you show that they are really important, and which we did, and we validated them in the human data sets with liver transcriptomes, which were acquired by us or public data sets or our colleagues. So concluding that, I wanted to say that the high throughput biology is so-called Wild West. And if you're a machine learning engineer or data scientist, you can try to adopt whatever methods you're using with your data in biological context, because there are new methods like every year and they become popular super unpredictably. There's a wide space for experiments. For example, random walks are being used in the networks based on network scoring, such as centrality, betweenness, and so on. But in this case, for example, I use them in biology relevant metrics, and you can try to figure out what is relevant for your question and try to apply that. You can apply that for new molecular targets, biomarkers, getting new insights if it's something not previously described. And ultimately, you will need to collaborate with somebody who would make sense of your output of your machine learning models, because unfortunately, often there is no technical metric you can apply, and there's only an universal answer whether the model result is good or not, is whether the biology of this model makes sense or not, the output of this model. So thank you again, and again, our team of Genomics UA, a bioinformatics organization in Ukraine, is gathering donates. So in case in case it's possible for you, I would appreciate if you would help us. Yeah, and that would be it. And I'm happy to jump to the questions.
Thank you, Alexander, for the interesting talk. Let me share the screen with the questions now. So the first one is, speaking about DNA, are related machine learning-based analysis typically generalized to both short and long reads, or there are separate implementations for these two cases?
Yeah, so I would personally consider that as the universal problem, like with the structure of the genes, because ultimately, like when you process or post-process the data from both short and long read methods, you basically get the same, from the data science perspective, the same readout, the same outputs or matrices you can work with. So I would imagine that you could apply these methods to both of them. However, it's really different because I would not assume that you can combine both of them really easily, because in long reads you kind of capture different type of information. You also capture, for example, certain DNA modification patterns that you cannot see with short reads, especially if you talk about nanopore sequencing. So depends on your question, it can be not really applicable or integratable with each other.
Okay, thank you. And another one is, what are the typical approaches for gene expression prediction in response to treatment?
Right. So there are papers on that, but I would not be focusing too much, to be honest, on that as like primary interest, because normally you would like to predict gene expression in two cases. First, when you want to impute the data, So, for example, you have missing values for the technical reasons. Your sequencer did to capture something. And the second one is when you work with methods which normally in RNA sequencing, you have this negative binomial distribution of your rates, meaning that you have a lot of zeros, like a lot of zeros. And sometimes you need to understand what is the true zero versus what was the zero for technical reasons for the sequencer didn't capture that. And then you might need to infer these zeros as non-zeros and predict possibly their expression based on other samples. Other than that, I would not do that. If you're interested in the later, there is this package which is called Magic, which is, I don't really remember how does it pronounce completely, but if you Google Magic RNA expression, you'll find that it helps with this. And for the second approach, KNN, key nearest neighborhood, would be just sufficient.
Okay, thank you. And one more question, the short one from me, regarding dimensionality reduction. Have you perhaps, it's very cool that you tried also like all these manifold methods. Have you tried persistent homologies? If you were on the first days of the school, there was actually a nice lecture about that. It's mainly used not for dimensionality reduction, but dimensionality reduction is also an option.
Yeah, I'm not sure I ever seen that using, at least in our field, in the field of this high-throughput biology. But in principle, there is no bad reason not to try it, and apparently, eventually it might even work in some fields, even if it's not primarily a dimensional reduction method of choice. It's so unpredictable which one would be the best in each scenario. It's difficult to tell. Many people just use UMAP because it's provided by the default packages, but if somebody tries and it gets effective, I would be interested to see.
Well, just a suggestion to you that if you decide to try other methods, that's also relatively new and cool method. Okay, thank you, Alexander.